Sorry about that. We ran out of time on the last one. We'll only go up to 15 minutes. So, we were talking about how mRNA has to be processed. And if mRNA is not processed, there'll be different enzymes that will chew it up between its trip from the nucleus to the cytoplasm. So we have to protect each end. One end as a 5' GTP cap, so guanine triphosphate. The other gets a poly A tail, poly many A's. And so you can see there's between 50 and 250 A's. So the longer the tail, it can last longer, produces more protein. So, in all, we again have DNA. It's a blueprint made into mRNA. So that was transcription. That is the piece we've talked about. We're going to talk about how that mRNA is then made into a protein. It's called translation, and ultimately into a trait. So, translation is nucleic acid language to amino acid language. Alright, so how does mRNA code from DNA? Code for proteins. So we're starting back with our DNA. So here's our long strand of DNA. We know it's DNA because it has T's in it. Then we have, so it has 4 different things, A, T, C, and G. Then we have mRNA, that T paired with A paired with U, so on and so forth. Everything paired together. We have 4 different differences, A, U, C, and G. Now, so that first part here, that's transcription. Now, how do we get to this? Our amino acids. There's 20 different amino acids, so from 4 base, so we have 20 different amino acids. How can you code for 20 amino acids with only 4 nucleotide bases? Well, first off, if you see the colors, every 3 nucleotides make 1 amino acid. And these aren't lining up real well, but you can kind of see. It's supposed to be over there, but yeah. And so the codon is every 3 nucleotides. So Crick, as in Watson and Crick, who found the structure in DNA, determined the 3-letter triplet, which is the codon. Why did the red bat eat the fat rat? So every 3 nucleotides. Nirenberg and Kona have determined that the mRNA amino acid match. So they added mRNA to a test tube of ribosomes, tRNA, and amino acids, created artificial UUU in mRNA, and found that that coded for phenylalanine. And from that, this figured out the genetic code. And so it's code for all life, and shows common origin because everything uses this. The code is redundant, so you'll get several codons for each amino acid, so all these codons go for ser. The third base is kind of a wobble. You don't always need it. The start codon is AUG. Which is methionine, and we have 3 stop codons right up here. And so to read these, so if I have CUA, I'd have 1, 2, 3. Basically, put all the lines together, you'd have Lu. So, why do we need a little bit of a wobble? Basically, if you look at third one, you have less of a chance of getting a wrong amino acid. So when we talk about mutations, that will become important. So how are they attached? Well, I didn't quite going in the right area, but AUG will meet with UAC. Those are the anticodons. That tRNA is going to bring the amino acid to it. So, going through this whole piece again, we're going to be bringing, that tRNA is bringing the amino acids over. So, the tRNA, cloverleaf structure, and it has the anticodon on one end, and an amino acid on the other end. In order to get that tRNA working, there's an enzyme which bonds the amino acid to the tRNA. When that's the amino acid tRNA synthase, we need energy. So ATP goes into AMP. The bond's very unstable, so it can reach the amino acid at the ribosome very easily. So the ribosomes facilitate the couple of tRNA anticodon to mRNA. The structure, we have RNA and proteins. 2 subunits, large and small. So there's 3 different sites, you kind of see them here, on ribosome. So you have the A site, which is the amino acid tRNA site, and it holds the tRNA carrying the next amino acid to be added to the chain. There. We have the P site, the peptidyl tRNA site, which holds the tRNA carrying the growing polypeptide chain. And we have the E, which is the exit site, and it's empty, and so that's how it leaves. So you can see, first part we have is initiation. Brings together the mRNA, ribosome, subunits, initiator. Elongation, as it goes across the mRNA, adding amino acids, and then termination of the end codon. And so we also have proteins. So you have a signal peptide. So these are the different proteins I could have. Here I have that signal peptide, and I could have different secretions, chloroplasts, all different uses for these proteins. So we have to do the whole thing. We have DNA. You can't really see it, but RNA polymerase. It gets the pre-mRNA, which has exons and introns, takes out those introns, adds a 5' cap and a poly A tail, and we get a mature mRNA that goes into the ribosome. And we have 3 different sites. This basically kind of think of it as the arrival site. As it brings in the next one, the P site that has the growing chain, and the exit site. It's being read 5' to 3', or tRNAs, amino acids, the synthase, putting those 2 together, and our growing polypeptide chain again. So, what about that gene? So, how we start it, we have the promoter, where it starts, enhancer, upstream, it tells it to go on and off, RNA polymerase goes on, and we start transcription. This is when we stop. So RNA polymerase goes through. We have introns and exons. The intron, that's where we start. and that's a stop. I'll turn it into a stop codon. We have our pre-RNA. The introns go out. We have our mature mRNA that has to have the GTP and poly A tails. Alright, so there's those, but what about prokaryotes? So, biggest thing, they don't have a nucleus, so things are a little bit different. Prokaryotes, DNA is in the cytoplasm. Eukaryotes, it's in the nucleus. Prokaryotes, circular. DNA, linear. Prokaryotes have naked DNA, whereas we have DNA wound on histone proteins. There's no introns in prokaryotes, and we have both. So, things are a little bit easier for them. They can actually go through, so here's the DNA, the polymerase, as it's making the RNA. DNA is already in the cytoplasm. We don't need RNA to be edited, and the ribosomes can read it. So there's ribosomes right on here. As mRNA is being made, ribosomes can start making the protein. It all can happen right there. During translation, there's no physical. So here's our prokaryote. It can happen right away, whereas eukaryotes, you have to go into the cytoplasm. Eukaryotes takes about an hour, and prokaryotes don't need any processing. So we always show it with one ribosome, but no, many ribosomes, we can make many proteins because it can all happen at the same time. Remembering from protein folding that it has to be folded up, primary, secondary, and tertiary structures, so line, pleated sheets, contorted, and the chaperones help that. And then, so here we have some modifications. Amino acids can be modified by adding sugar phosphates. Enzymes can modify the different ends, and so for instance, we could have insulin that's connected. I don't know why this is important. And so we have this guy who suggested that there's phenotypes through different and he started the one gene, one enzyme going in. Found that when an enzyme goes out, that could be an inherited disease. And for instance, black urine, that is a defect in an enzyme that breaks down amino acids. So, mutations. Let's see if I can talk about these quick. So you have point mutations, which is just or a substitution, where a single one is changed. So there's different things that could happen with this point mutation. It can be silent. Nothing happens, so we have the same amino acid. We can have a missense, which will change the amino acid. So I went from gly to ser here, and I can have nonsense. Oops, stopped too early. So there's a point mutation that leads to sickle cell. Oh, sorry. So the type, if you look, amino acid is changed, and so we have a missense mutation. And that changes our cells from being nice little round ones to sickle-shaped. Other ones, the frame shift is when everything is missing, so I have 3, 3, 3. Oop, if this is missing, it moves that amino acid over, and you can see everything then shifts. This can be caused by insertions or deletions. More damaging at the beginning than at the end because you're getting more amino acids. So, DNA and DNA. And